Hi, today Jeana and I are presenting a paper published in the Protein Society Journal in 2018. The title of this paper is Flavin Oxidation in Flavin-Dependent N Monooxygenases. This paper was researched and presented by Reader Robinson, Catherine Clanker, Pedro Rodriguez, and Pablo Sobrado. To begin, we're going to discuss the background and information of flavin-dependent monooxygenases. These compounds are small iron chelating compounds that perform many redox reactions such as hydroxylations and reductions. These reactions rely on NADPH as an external proton donor. Because of this, flavin-dependent monooxygenases are important biological catalase. Because of this paper explored the siderophore side chain of these monooxygenases and the efficiency of the hydroxylation reaction based on the amino acid bound to the siderophore side chain's active site. This paper explored the use of these compounds as drug targets since they are involved in the biosynthesis of hydroxylate functional groups in siderophores. The goal of this research article was to determine whether or not specific protonation states for ornithine and lysine amino acids were required for the hydroxylation reaction catalyzed by the side chain siderophore A. The authors focused on two main studies involving pH and isoelectric point to determine which amino acid would be most efficient in the hydroxylation of the SID-A side chain.
The reaction catalyzed by SID-A starts as the enzyme with the oxidized form of flavin attached. Flavin is reduced by NADPH to form the reduced FAD-NADP plus complex. This complex reacts with oxygen to form the C4-hydroperoxyflavin. Once this happens, ornithine can bind and is hydroxylated at its N5 atom. Following this, water is released from the hydroperoxyflavin to form the oxidized form of FAD, or the hydroxyflavin. The products of this reaction are NADP+, water, oxidized ornithine, and the hydroxyflavin, which remains attached to the enzyme. This completes the reaction as discussed in the article. The pH profile of siderophore A was determined by measuring the rate of oxygen consumption, while the NADPH concentration was kept constant. The results of this study showed that increasing the pH also increases the kcat value of siderophore A with a pKa of 7.0. Kcat is the turnover number, or the number of substrate molecules converted to product by the enzyme per unit of time. These numbers show that the ornithine amino acid was converted efficiently. The amplitude of the kcat over km shows a similar trend with increasing values as the pH increases with a pKa value of 8.0. Thus, as the pH increases, the apparent Km for ornithine decreases, indicating high affinity for the substrate. These results show that the specificity of the enzyme for the substrate is greater at a higher pH level, which indicates that it is the deprotonated state of ornithine that is favorable for this reaction.
This article also studied the effects of isoelectric point on the hydroxylation of siderophore A in the presence of no substrate, lysine, and ornithine. Isoelectric point is defined as the pH value where a particular molecule carries no net electric charge and is neutral. The isoelectric pH profile for flavin oxidation was determined in the absence of substrate as well as in the presence of ornithine and lysine. This measured the efficiency of the hydroxylation of the siderophore A in the flavin monooxygenase. To begin, the absence of substrate was determined and their pH profile was considered. The pKa was determined to be greater than 9 in a water solution where the rate constant for flavin oxidation increases as the pI increases. Because the siderophore side chain is unstable at a pKa greater than 10, This is not a favorable process because the high pKa is needed to hydroxylate the side chain. Instead, we considered the pI profile in the presence of ornithine to determine the ionizable groups important for hydroxylation. This graph shows a significant shift in pI compared to that of the pI in the absence of substrate. This reduced the pKa value from 10 to 6.7. Even though lysine will bind to the active site of siderophore, the hydroxylation is not efficient, which can be seen in the PL profile conducted. In the presence of water, the pKa value of lysine is 7.7, which is considerably higher than the pI profile in the presence of ornithine. These studies show that the pKa found for ornithine is more cohesive with the pKa needed to stabilize the siderophore side chain and thus stabilizes the hydroxylation reaction at the active site of the siderophore side chain. As we can see from the obtained pH and pI values, the isoelectric points of both lysine and ornithine are substantially different. The PL profiles found in the presence of siderophore and ornithine are 6.7. Comparing this to the known value of pKa value of ornithine at 7.1, we can concur that this reaction readily takes place because the siderophore side chain is stable at this pKa. This indicates that the amino acid group needs to be deprotonated for optimal activity and hydroxylation. However, while in the presence of lysine and siderophore, the pKa is 7.7. and hydroxylation occurs less than 10%. This is because the pKa of 7.7 as compared to the known pKa of lysine of 9.47, lysine would not be deprotonated and hydroxylation would not occur because the lysine would carry a positive charge and not readily bind to the siderophore side chain.
In conclusion, this study shows us that in the hydroxylation of siderophore and flavin-dependent N monooxygenases is most efficient when ornithine is present as compared to that of lysine. Because ornithine is more readily deprotonated and neutral at a pH where siderophore is stable, binding to the active site will happen readily and hydroxylation will occur, producing the necessary hydroperoxyflavin complex. If lysine were used, hydroxylation would not occur as readily and hydrogen peroxide would be released as a product. Thus, we can conclude that ornithine is the preferred amino acid for flavin-dependent N monooxygenases. To finish our presentation on this article, we would like to discuss why this study is relevant in the study of biochemistry. Flavin-dependent monooxygenases catalyze a wide variety of chemo, regio, and enantioselective oxygenation reactions. As such, they are involved in key biological processes ranging from catabolism, detoxification, and biosynthesis to light emission and axon guidance. This makes these molecules wildly important in the treatment of medical conditions and stabilization of various disorders. For example, hypertension, also known as high blood pressure, is found to be regulated through the use of these flavin-dependent monooxygenases. These compounds trigger biological cells to release a hormone necessary to counteract the increase in osmotic pressure and peripheral resistance. Thank you for your time. We hope we have informed you on this important study of biochemistry.